Yeah, like I said, like Phil said earlier, I'm Will. I work for Eli Lilly. I work on methods and tools for clinical statisticians, and I'm really excited to talk about the newest stuff in crew and targets, as well as talk about simulating clinical trials specifically. Particularly excited about this workshop because I get to talk about a kind of workflow, a kind of pattern that my team and I have been working on internally for a number of years. And this is really where we're coming from in terms of these large pipelines that we're doing. And I get to share all this context with you as well as the tools that are doing these things. So um, with that, I'll be starting briefly with clinical trial design and simulation. Then we'll move on to an example simulation project to show how I would express how my team and I express simulation projects in our code to support the trials that we're working on. And next, I'll go into targets and how that fits together with all this. And then crew, which is a new package. I've been working on it all year. And Charlie Gao has worked. He's a collaborator of mine externally. He's been working on the back end of crew packages, Mirai and NanoNext that I'll get to. And we've been working together on this for the whole year. And this is the first public facing presentation that I'll be giving on crew. So I'm really excited for that. So to start off, many of you are familiar with the drug development process as a whole. And this certainly is not going to be a course on clinical development, but I will just start off with some orienting context here. In the drug development process, we start off with thousands or tens of thousands of possible compounds and chemical pathways that could lead to treatments for diseases. These the possibilities are just overwhelmingly enormous at the beginning. And in drug discovery, there are studies and assays and analyses with all kinds of omics data to select the most promising candidates for the compounds and chemical pathways that work on. That gets funneled down to other kinds of non-clinical studies like animal studies with a subset of those molecules. Very few of those make it into human trials and are studied for safety and efficacy on humans. And then the most promising of those go to regulatory review if there's enough evidence for the safety and efficacy. And those become therapies that are the ones that pass review or become therapies that are administered to patients in the real world. So I think many of you have seen this, and my colleagues and I work on the clinical trials space of this. So this middle section in this overall drug development process. And there, the funneling effect goes the opposite direction in terms of the number of patients who were involved in the study. And so when a candidate drug enters the clinical program, there are very few people who are given the drug and in very, very small doses. Once more and more evidence of safety is established, and then as more and more efficacy is established, the number of patients involved in phase 2, phase 3, and beyond increases. And so as we grow more and more confident in the safety and efficacy of the models that we study, the more and more real patients are involved in these studies. And that's important because there are an enormous number of risks and some key scientific questions that we need trustworthy data to substantiate. In terms of scientific questions, it's most often going to be things like, is this compound safe in humans, and is it well tolerated? What is the maximum tolerated dose? Under what conditions can this compound provide benefit? what indications can this compound treat? Which patients can benefit the most? How efficacious is this compound compared to existing therapies? As well as questions about PK/PD, dose response, and the underlying mechanism of disease. As we answer all of those questions, we have to absolutely must protect the safety and the well-being and the privacy of patients. That's what makes this whole process of designing a trial a very careful, methodical, and conscientious process that goes on for a long time and has a lot of quantitative evidence to back it up. And part of that quantitative evidence to support the design of a trial is simulation. Not everything in a trial needs to be simulated, but it's extremely useful for very targeted questions and questions where we need quantitative evidence to justify a design decision or characteristic, but that we don't necessarily have a whole lot of data just yet. Lack of data is why we're doing the study in the first place. So this could be to compare different decision rules in a trial. Combined with available data, we could use simulation to decide which patient populations to design the trial for, which indications to choose, and which design characteristics, like the number of interims and when they should be. Tuning parameters such as sample size, number of doses, to set up an algorithm like response adaptive randomization. That's a really complicated, potentially rewarding procedure that simulation can help tune. But again, of course, not everything needs to be a simulation. If it's just a frequentist sample size calculation, then maybe simulation is an overpowered exercise. But if the models in the statistical analysis plan are really complicated, for example, some Bayesian models that some of you may be working on right now, then you might use a simulation to select sample size among other characteristics that you're interested in. So how simulation for a clinical trial works in the experience of myself and my team is it's an exercise where you draw a set of hypothetical patients from a probability distribution. And that's the first step. And that distribution depends on the different assumptions that you might want to put in place as far as what is the available data so far say, and the mechanism of action so far say about what the response to this endpoint should be, or should be in a worst case scenario or a best case scenario. Using that information, you would write some code to simulate some patients, and that could be one or more data frames of virtual patients. And those will have baseline characteristics, and those will have outcomes, which could be any combination of continuous and dichotomous endpoints. The next step is to analyze those outcomes with the statistical models of your trial. So if you're running, whether you're running a frequentist model or a Bayesian model, you would analyze the patients that you just simulated with our code and report final decisions like posterior probabilities and p-values that'll let you determine, it'll help determine efficacy, whether the trial that you've just simulated declared efficacy or safety, or where it didn't meet those milestones. And then that's just one simulation replication. And the exercise in simulation is to repeat this process for thousands of times for different hypothetical scenarios to really get at what we believe to be about the conditions under which the drug truly works or the drug doesn't work. So we would first posit maybe a scenario where there's zero efficacy, and then simulate the trial and count the proportion of times that the drug declares success. And those are going to be your false positives. That's valuable information, and that trial may be may suffer from type 1 error that's too high in that case. And reverse, you might want to posit a scenario where where the drug is some effect size determined by a competitive landscape analysis or existing data, or some percentage better than an existing therapy. Simulate under those conditions and see how often the trial declares success when it's simulated. So that'll get you the power of that study. And it's good practice to simulate some scenarios in between to see what would happen in sort of the grayer areas. Now, this process is straightforward to explain, but it comes with some pretty big challenges that we've struggled with historically. And there's really intense computation when it comes to especially complicated, highly adaptive, innovative designs where there's a lot of complicated bits and pieces happening operationally. If there are a lot of interims, or if there's a complicated randomization procedure, different stages of the study, or if multiple studies are simulated. So phase 2-3, seamless phase 2-3, for example. Not only that, the models are quite complicated and challenging, especially if you're talking about Bayesian models, especially longitudinal disease progression models, as in neurodegeneration. Those things will not only present a challenge as far as writing the code, the computation time will be quite long. And amid all of this, it's very common that the work environment is quite hectic, and there's a lot of decisions being made mid work stream, and design elements changing because of factors that may not be necessarily quantitative but still need to happen. It may be an early stage where the whole study team is working out its priorities. Maybe the competitive landscape changes while you're working on this, and so there's this need to be able to turn on a dime and to turn results around quickly, which includes changing existing scenarios, adding scenarios that are requested, changing fundamental aspects about the design, things that may need to be recoded. And the workflow and the pattern that I'm going to describe is going to address this in a modular way and in a way that scales, and that we found very successful, and that we've been using on it. The techniques that I'm going to describe are foundational to our simulation workflows, and really have been since the days of COVID, and afterwards. So but the trial right now I'm going to focus on is very simple. It's let's say a randomized, controlled, parallel, non-adaptive phase 3 study with a non-longitudinal analysis. We're going to randomize with a 1-to-1 allocation. So half the patients go to a drug after screening and half to placebo, all in a double-blind fashion. The main outcome is continuous, and we're going to assume, so we don't confuse ourselves, that a higher score on that endpoint is a more efficacious outcome. And we're going to run a frequentist analysis that declares declares efficacy if our p-value is less than 0.05 in a one-sided hypothesis test. Now, this is a trial that we could probably definitely just do a power calculation, a sample size calculation with a back of the envelope procedure. We don't really need complicated code to simulate this trial, but it's useful to start somewhere, to start small for the sake of demonstrating the tools. And I'll point out in the R code where you can add more complicated operational bits. This is a successful pattern for more complicated trials, but just so we don't get bogged down into the clinical design aspects for this presentation, I'm I'll be focusing on a really simple study today. And the goal of the simulation, and the goals, by the way, of different simulations, it's going to be different. It's not always going to be very sample size, calculate power and type 1 error. That's very much going to depend on a set of motivating questions that should be explicitly clarified in advance. And these simulations should be very targeted because of the amount of effort that's required to set them up. But the goal of this one is a simple sample size calculation. And it's worth stating this explicitly. We want to find the minimum number of patients to demonstrate superiority to placebo with 90% power and controlling type 1 error at less than 5%. Before I go on and start talking about the code, yes, to respond to some of the questions in the chat, yes, there is a Posit Cloud workspace. Apologies that I didn't put it in the chat to start. I did set up a cloud workspace that you can feel free to follow along. The cloud workspace is it matches the example GitHub repository that I put in the chat earlier before the presentation was started. Feel free to follow along if you want. It's really convenient for just getting up and running with the code without having to install any packages yourself, or even R for that matter locally. But if you want to just listen along to me talk and to go to the live demos if we have time, that's great too. So that being said, another question is the alternative hypothesis here that treatment efficacy is superior to placebo. Yes, that's the alternative hypothesis, and the null hypothesis is that the treatment efficacy is not superior to placebo. This is yeah, or just equivalently that they're indistinguishable for the purposes of setting up this kind of test. I mean, there are distinctions. Of course, basic fundamental statistical considerations as far as what your null hypothesis actually is. But for the sake of this test, you'll get the same operating characteristics, and the main takeaways are going to be the same as far as this example and the tooling. All right, so we have an example trial. We know why we're designing it this way, and we want to express this in terms of code so we can run a simulation to find that optimal sample size. Before we begin, I'm going to recommend that we take a function-oriented approach here. So R has a lot of great features when it comes to functional programming. The concept of functional programming gets very deep, but at the very basic level, to use a chain of functions to express the analysis that you're doing is extremely valuable and extremely clarifying, and makes the analysis clean and modular, done carefully. So arguably, a data analysis is a sequence of transformations. We begin at a set of scenarios that posit our hypotheses about drug efficacy. And then we write functions to transform, for example, a scenario into a simulated data set, because one depends on the other. And we have another function to analyze that data set, and then a function that composes both to simulate and analyze together. These are shorthand, these functions, to make our code much easier to read and much cleaner and easier to maintain, and just very, very clarifying and nice to work with. And typically, I mean, if you've worked in a language like C before, or C++ functions may come a bit more naturally, but in R, sort of the things that you would write a function for in this case would be to simulate a data set, or analyze a data set, or post-process an analysis to display results. those are some of the main cases you might write functions for. And then if any of those functions become very big, then it's a good idea to break them up just into smaller functions that you can compose together for maintainability. In our case, our simulation function is going to look like this. So we have a simulate dataset function, and our mean drug response is going to be some number, and our sample size is going to be some positive integer. We begin by simulating a bunch of patient IDs. We designate the drug arm. We keep track of which patients are assigned a drug and which patients are assigned placebo. We simulate responses, virtual responses for the patients who are taking the drug with this mean. And then we have a different mean for all the placebo patients who we simulate. And then we get a nice tibble in our return value that shows the patient ID, the study arm that each patient is in, and their response to the primary endpoint of the study. And we have in our data set, it's this nice tidyverse-friendly data frame with one column per field that we're interested in, and one row per patient. If we were simulating a longitudinal data set, we might have one row for every patient and study visit, or per every patient and time point. That's also a very common pattern as well. We have another nice function to analyze a data set, and we take in, for example, this data set argument is assumed to be the data set that we just simulated. We analyze this with just basic linear model with the LM function. And we conduct a hypothesis test to get a p-value using the t test statistic that we got from the LM function. And I'm assuming here that we have enough patients to use the normal approximation. In more serious scenarios, you might use different distribution for your test statistic and apply degrees of freedom as from the Satterthwaite method, trying to keep it simple here and assuming that we have enough patients in this phase 3 study so that those normal approximations are valid. The choice of analysis and the choice of modeling, it's seldom this simple, and it depends on the specific study that you're simulating. And a lot of thought and care and research goes into developing the models and the statistical analysis plan. At the end of this, you get a nice data frame again, and the result has one column per metric that you might be interested in. For example, the p-values that decide whether we're going to declare success or not. These could also include posterior probabilities, effect sizes, quantiles, posterior predictive probabilities of success in a later study or in a different study. All these you might return in, let's say, a one-row data frame or a very small data frame for the study, and the result's going to have maybe one row, maybe a few rows, but these are all going to be in a nice consistent format where we can bind together multiple simulations. The simulate trial function composes both of these functions together, and so we're going to simulate a data set using a given mean response in the treatment arm and sample size, and we're going to analyze that data set with the simple model that we've specified. And so that's the R code that expresses this simulation. If this were to become more complicated operationally, then you might have a more complicated version of simulate dataset. You might have different groups of patients in different stages. You might have a response adaptive randomization where there are analyses at each of several interims along the way. In that case, you might not be able to compose the data set and analysis functions together as cleanly as we were here. You might combine elements of both and simulate some patients then analyze those, then simulate some more, and then analyze those, and so on. But either way, you'll end up with a nice top-level function that accepts very simple inputs and delivers very simple outputs and runs what could be a really complicated, highly adaptive, maybe even highly Bayesian trial in the body of this function. And we're going to run this simulate trial function under a different set of scenarios. And usually this list is quite longer. There are different scenarios about the disease state or patient population or decision rules, and the strength of the drug and sample size for that matter that you'll want to be considering. For this, just to keep it at a nice simple level, just for the sake of this presentation, we have 4 different scenarios with 2 different sample sizes that we're going to test out, and 2 different scenarios about drug efficacy. We want to consider a efficacy scenario where we assume that the drug response is fairly strong. So that's going to have a higher value in this case of the mean drug response parameter that we saw as a function argument. Then we're going to have a null scenario where the drug and the placebo are assumed to have the same response. So for example, if your null hypothesis is that the drug and placebo are the same in terms of their outcome. So we're going to run simulate trial on each of these sets of parameters for 1,000 or maybe even a few thousand reps. I would definitely go several thousand reps per each of these scenarios. In practice, maybe you only start off with a small number just to get the computation up and running. But we want to reduce simulation error to be able to trust the results here. And then each simulation replicate is going to return a p-value, and we count the number of p-values in each case the count number of simulations in this case that have the p-value less than 0.05. That's the frequency of efficacy. And then depending on the scenario that you're in, the efficacy scenario, this could either mean power or it can mean type 1 error. And again, the goal of this particular simulation, not of all simulations, but of this one that we have here, is to figure out which is the lowest sample size among that we've considered that delivers greater than 90% power, greater than or equal to 90% power at less than 5% type 1 error. So at this point, we have a clinical trial that we're going to design, a simulation project, and we have some R code to express one single replication of the simulation. And so this may or may not take a while just to run a single replication, but in practice, no matter how long it takes, you can usually scale down for the purposes of development by running, if you have a Bayesian model, maybe you choose fewer MCMC iterations. Either way, all the functions that I've showed previously are developed as you would develop any other piece of R code. You would experiment with them interactively to make sure that you're reasonably sure that they run without any obvious errors, if they produce output that looks okay. You're not going to catch all the errors necessarily, because some statistical mistakes can happen, and you may not see those until you simulate for thousands and thousands of times. But you at least want to do all the things that you may already know how to do to get those functions in pretty good shape locally. Then comes the point where you might have to scale up the computation, run these simulations thousands of times to get reasonably trustworthy operating characteristics. And when you do that, this is where the computation usually becomes extremely difficult and hard to manage. And this is where I'm going to transition into targets and to describe how targets can help with a project like this, and why my team and I have been using targets for years to successfully manage and update our simulations internally. Just checking the chat real quick here for new questions. Thank you, Eric and Lucas, for answering this. So yes, for as far as setting the seed for a simulated data set, this is important for computational reproducibility, to make sure that not only that your simulation replications are statistically independent, but also that that they're reproducible. So successive runs of the same analysis will produce the same result, which is important for both regulatory reasons and to help you debug, troubleshoot anomalies, et cetera. And targets does have a way of setting seeds for individual simulation replicates and individual steps of the analysis. Parallel computing in R in general is difficult, as in the sense that it's difficult to construct your random number generators in a way that preserves statistical independence. And I've made recent updates to targets to make that a bit safer. Specific information about that is on the discussion threads, and there's a random number generation section of the user manual that talks about all that. Okay, moving on to targets. First, I'll say that targets is not just a simulation tool. It's for, we use it for clinical trial simulation. We use it for Bayesian data analysis, especially to run the kinds of simulation and analysis studies that we use when we're developing and testing a new Bayesian model that we're using internally. We've been running more and more Bayesian analyses, not only for just ordinary studies in general, but for master protocols, to do historical borrowing, to do disease progression modeling and neurodegeneration. Lots of Bayesian models over the last few years. We use simulation to make sure that we are to explore the behavior of this model, under certain kinds of data, to test our assumptions, and to make sure that we trust the implementation of our, for example, stand code and Bayesian packages that we develop. So there's a lot of simulations that we run with that are both for individual trials and for model development. Both cases come up a lot, but there are more. Network meta-analysis is computationally intense, and targets can manage those kinds of computations. Those data sets for network meta-analysis come from data curation exercises usually to assess the competitive landscape of an indication. And there are sophisticated Bayesian models that run to everything from hierarchical models and beyond to compare studies in a very indirect but principled way. Things like PKPD, statistical genomics, where data sets are enormous, machine learning, and other applications come up a lot that are heavy computations that are done in R. And for this kind of exercise, the usual tools and techniques that are historically taught can break down a lot of times. An example is one pattern that's often taught where an analysis project is organized as a series of literate programming reports, where you have maybe a Quarto or an R Markdown report that starts with setting up some of the initial conditions, another to run the next stage of the work, maybe simulating or gathering data, another to run the analyses, and another to produce summaries and visuals and so on. And each report might have an informative name and might have a numeric index to indicate which order the report should run. For anything even a little bit complicated, I've seen this pattern break down over and over again, especially for the large computations that I'm talking about here. So what this looks like in practice is not a straightforward one to the other. You have to manually manage output files. You might have to write output files that the next report produces, and so on. And you'll have to manage, data caching yourself. You might have to insert reports that you weren't intending to write at the beginning and insert them somewhere in the middle with an awkward index like 0 2a. And also the computational demands are another factor that really messes with this kind of workflow. Not only is that if you're manually writing parallel code in R and you insert it in one of these reports, it's going to look really complicated because the trial that you're simulating is already really complicated. And then code to run tasks in parallel just obfuscates the technical content of the analysis. You really want to have the code that expresses the operational and statistical characteristics of the trial separate from the code that runs things in parallel, that schedules things, and so on and so forth. Not only that, because of the computational demands that a simulation or genomic study or other kind of study requires, you might find yourself, especially in a fast-paced clinical design scenario with a lot of cross-functional collaboration and moving parts, you'll find yourself in a situation where you'll have to make a change and rerun a simulation project. And when teams want quick answers to questions that may on the outset seem quick, but that may require you to run the simulation all over again or to tweak this or that part. And it's easy to be faced with the question of, do I really need to run this from scratch all over again? And without a pipeline tool, the answer may be yes. And you may want to skip certain pieces of the code to save time in those fast-paced scenarios. But it's hard to look back and see, well, what needs to be updated and what needs to rerun and which results can we skip to save time. And as soon as you start thinking about that without a tool to guide you in a principled way, then the project becomes too out of sync and too messy to be truly reproducible and trustworthy. You would really want to have all your results up to date before sharing them with the team. So this is where pipeline tools come in, specifically make-like pipeline tools like targets. The main contributions of those tools are to orchestrate all the moving parts, all the different kinds of simulations that you'd be running, and to skip targets or steps of the computation that are already up to date. There's a very principled automatic set of rules that the targets package goes through in order to accomplish that. And except for some edge cases, you'll be able to say, well, targets told me that all my results are up to date with the code. That must mean that all the code and the commands and steps of the analysis in this graph really didn't change. But maybe some do. Maybe you have a scenario of simulations that's outdated and needs to be run, but maybe there are other scenarios that needed to be that can be skipped to save time. And the stuff that does need to rerun again, targets with the help of crew can run computation in parallel. And also the output data is in a nice, neat data store behind the scenes. And there's abstractions in place that you can interact with that look and feel like you're just operating on objects in R. And everything looks and feels like you're just operating in the comfort of your own R session, which is why targets is it's really nice. It's idiomatic to R. Again, it looks and feels like you're just working in your local R or RStudio session for the most part. It also encourages. It's a very opinionated tool, and its opinions are helpful. It encourages good function-oriented programming habits, which is why I started with the simulation code that I showed before with a very function-oriented style, both because targets nudges users to do that, and because it's just a really good idea for data analysis. It's got make-like properties like automatic dependency detection in terms of fitting the pieces of your code together. It's got behind-the-scenes data management, and there are a plethora of distributed computing backends that it can connect to. So there are resources already in place for getting started with targets. There's this 4-minute video that I'm linking to here. That's a very simple project with 3 or 4 steps in the analysis. And you can begin by just developing functions for this data analysis in the usual way. And then using the use targets function, it's very smooth, easy transition in order to get from just a function-oriented data analysis project to a targets pipeline. And whether you're just getting started out or want to look up clarifying details about this or that feature of targets, there is a documentation website, and there's a user manual that can go through all of the user manual goes all through all the major topics, and the documentation website has a bunch of links to useful resources and references for all the functions. And so the documentation is quite extensive, and it can help you whether you're just getting started. There are friendly materials like a walkthrough chapter in the manual, all the way to advanced features in distributed computing. So to get started, you'll begin with the simulation code. It's common practice to put your code in a folder called R. This is also common practice whether you're developing a package or a Shiny app. But we'll say that there's some place where your simulation code that you've already written by hand lives. And then on top of that, the difference between that and a targets pipeline is a targets pipeline has a special targets.r file. And that's created initially by the use targets function, and then you'll modify that script by hand. This targets file, by the way, that I'm highlighting, this is similar to a make file. Targets is kind of like make for R. Don't worry if you don't know what make is. It's a low-level tool to compile C, C++, or Fortran programs. Been around since the 70s. But if you happen to use make before, targets derives a lot of its operating philosophy and configuration from just the general concepts of make. And this is like a top level configuration file. If you are writing, let's say, a Shiny app, and you know that you have a central app.r file for your app, this targets file is kind of like your app.r for a targets pipeline. It's this central file that makes a project really a targets project. And I'll go in detail about how that works. After the use targets function creates that, you'll, like I said, edit it by hand. It's a really good idea to write a small test pipeline just to make sure things are working correctly before you scale to thousands of simulations or hundreds of workers on a cluster or on AWS Batch, for example. There are tools in targets to inspect the structure of the pipeline. There are functions tar manifest and tar network, tar viz network, and other sorts of functions to help you do that. The function tar make runs the pipeline, and that's the one that actually executes the analysis that you planned. And then there are functions like tar read and tar load to load results. So a bit about this targets file. When you first generate it, it'll be a file that's actually mostly comments with a little bit of R code in it. The good practice is to read those comments and follow the directions and follow along. By the time you adapt it to your pipeline, it may look something like this. It starts out by loading the packages that you'll need to just define things. this is kind of like setting up your R session. You just, and in this case, you'll load targets. You might load other packages like tarchetypes or other similar packages that'll allow you to do some setup. Then you'll set some top level configuration options, and I'll say the packages that the analysis needs to actually run are dplyr and tibble. Those are going to be the packages that the simulate trial function is going to need. The tar source function is really convenient. It just sources all of your function scripts in the R folder. So the simulate trial function and other functions are going to be loaded into your session with this tar source function. Everything in lines 1 through 7 is the same kind of setup that you would do for your local interactive R session if you're just planning an analysis. This is very idiomatic to R. This should look and feel like you're just working in your own interactive R session. When the pipeline actually runs, it's going to do this in a different process. But this is the look and feel of it. And then the last thing in the script is a list of target definition objects. And this is kind of like defining, if you were to define a Shiny UI or a Shiny server, if you're writing a Shiny app, this is kind of like the same basic R code that would specify that specifies a configuration for this for a pipeline. So each target declared with the tar target function is a step in the analysis, and it requires a name, and you would write a symbol for that. This gets quoted and interpreted as a symbol, and an R command to run. This could depend on and will depend on the functions that you've written for simulation. It calls simulate trial. When this tar target function gets run in practice, it'll actually quote both of these arguments. It'll take this code, and it won't run it now. It'll run it later when you execute the pipeline. So we have, for this example, small demo pipeline, just to make sure things are working properly, we have a simulation under one scenario, a simulation under another scenario, and then a target to combine the simulations that we've already done. And targets understands, by way of static code analysis, all these codes and all these, how all your functions, all these commands fit together. And so if we run tar viz network, you get a dependency graph of all of the functions and objects and targets in the pipeline. So for example, this results target depends on simulations 1 and simulations 2, because the command for the results, as you can see in tar manifest, depends on these symbols, simulation scenario 1 and simulation scenario 2. All this depends on the code that you've written, because these simulations call the simulate trial function as well as these upstream functions. So what makes targets declarative and nice is that when you're setting up your pipeline, you only need to worry about the functions that you write and the individual commands for specific steps. And then the package will figure out how all this fits together in terms of a left to right flow of the analysis. And then later on, as we'll get to, how to run these things in parallel. So the tar make function runs the pipeline from start to finish. It runs the correct targets in the correct order. And the results are in a data store. And this is what the data store looks like. If you do a dir tree from FS on this, this will be a folder called _targets. It'll have a bunch of metadata, and then it'll have one file per target that you have in your pipeline. Unless you're using cloud storage, and I may get to that, in which case those will be on an S3 or Google Cloud bucket. But you won't have to, regardless, you won't have to really worry about where these things live. All you need to do is call a function like tar read, and it'll automatically go get the file and read it into your R session. And this is one of the best parts about targets, if you change a command, for example, a sample size and that you're running with, the targets package will detect that change. It'll say that this target needs to rerun, and possibly anything downstream. And so when you run tar make, it'll automatically run the simulation that you changed, skip everything else, and then bring the whole pipeline up to date that way. Likewise, if you change a function, it's sensitive to changes in the body and arguments of any functions that you define, and it's aware of the results that depend on that. So if you change a function here, then in this case, all the other targets need to rerun. And conversely, if nothing has changed, then targets may say all of your functions and all of your targets are already up to date. And this is concrete evidence of reproducibility. It's evidence that your results are synchronized, the output matches the code that you supposedly ran it with, and you can start sharing the results if the statistical issues are also correct, and you can work out downstream discussions about the design of the trial with the team. So that's basic targets functionality. Now I'm going to transition into things that are built on top of targets, and that targets depends on, which can accelerate and expand the capabilities of the analysis. So, as I mentioned before, by default, targets saves its data locally, but you can configure settings to store the data in, for example, an S3 bucket or Google Cloud bucket instead. And all that requires is to declare a repository argument, which is either AWS, GCP, or local, and then give it more details about what bucket that you're selecting, where in the bucket this project lives, and a bunch of other options if you like. Also, that as of some recent updates to targets, even the metadata is stored on S3 or Google Cloud if you select a bucket at the level of tar options set, which will allow you to, which really opens up different kinds of deployment options. You can run a continuous integration workflow, which pushes to a bucket and then pull that metadata in a local session and interact with it and check on the progress of the pipeline from the comfort of your local computer and local R session. And there are use cases for this that I haven't quite fleshed out yet, but really eager to, because there's in distributed computing scenarios, you're usually on a different kind of environment, like a cluster, and all your interactive development and exploration and troubleshooting. It's much easier when all that's locally, and the new features around metadata on the cloud can let that happen. There's a mechanism called target factories, which we've been using quite a bit, not just for simulation, but for Bayesian data analysis. There have been external examples in PKPD as well. This is a really powerful pattern to create shorthand for different sections of the pipeline, and to abstract away a lot of complicated features like dynamic branching, so that end users don't really have to. So if you're a package developer and want to extend targets, this is a really useful technique. How this works is you write a function, maybe standalone, maybe in a package, that accepts a very simple set of inputs and defines a list of target objects as you would in a targets.r file. And then when you go to use it, you write this target factory instead of the tar target function. And instead of one target, which is what this looks like, you'll get several. So in this case, the target factory function doesn't have to be named that. It can be named other things that suit the use case. But this will give you 3 different targets in the pipeline. And that's you can define multiple instances of this target factory. Maybe if you supply it with a different name or a different, for example, data set here. And this mechanism supports Bayesian data analysis, and it also supports literate programming in the Tarchetypes package. So Tarchetypes has functions for general kinds of repeated tasks that you might do in targets. And it has a bunch of built-in useful target factories. One of the mechanisms here is one of the patterns that it supports is if you want to summarize and narrate and talk about your results from the pipeline in a Quarto or R Markdown report, the tar Quarto and tar render target factories let you do this very easily. And the idea is to have a report that really doesn't do much work other than reading targets that were previously computed, showing them in these code chunks. Maybe you'd have some text in that's above or below this code chunk to describe what these results mean, where they came from, et cetera, et cetera. This is really nice for collaborating among different statisticians and even to study teams about the results of a simulation study. And what this looks like in our example for this report, all you need to do is to add a tar Quarto statement for this report at the end. So this is the name argument called report. This is quoted as a symbol, again, like the other targets. And then you would give it the path to the Quarto file. And this pipeline now looks like the pipeline you see here. We have this new target report that has never been run. And so when we run tar make, it skips all this potentially expensive simulation work that's leading up to this point. It runs the report because it's the only target that's new or that hasn't changed since last time. And this is usually very quick. Again, this report shouldn't really do any non-trivial work of its own. It should just be pulling in, concisely pulling in the results of previous steps in the pipeline and sharing them amid explanations that clarify what these results mean. And looking at this report, this is what we see. We don't really have we don't really have much here other than just a display of the results from the 2 simulation replications we've run so far. And this is not only possible to continuously run this report as part of a pipeline. When you're writing this report locally, as long as these upstream targets exist in the data store, you can open your source file in the report in, let's say, your RStudio IDE or VS Code IDE, and run the report locally, and just use develop the literate programming report just like you would normally outside the pipeline. It's a really nice pattern and a nice combination. So up until this point, we've only run one simulation replication per scenario, and that's good for establishing the structure of the pipeline, running it quickly, making sure that it works before we scale up the number of simulation replications, and before we may place in more computational demand on each simulation. So, for example, if we're running a Bayesian model, we would want to add enough iterations and MCMC chains to satisfy convergence diagnostics and to be reasonably sure that our model is fitting the right, approximating the correct posterior in a correct way. So we're going to now scale out to all 4 of our scenarios. And for this phase of the pipeline, we're going to run each scenario 1,000 times. In practice, we're going to probably want to scale this up to even more simulation replications. But that's the scale we're going to attempt right now. And this would ordinarily be a pipeline where we would do manual static branching over scenarios and dynamic branching within replications and a batching structure to optimize some computational trade-offs between overhead and and speed, etc. But we have this really nice target factory from the Tarchetypes package in order to do this. And it's called tar map rep. We've been using this target factory for years, for a long time, to do this very repeatable and this very common kind of simulation structure, where you have a data frame with one row per scenario and one column for per parameter. And each of those scenarios defines some kind of simulation that you want to run thousands of times. This is so common that this target factory is really one of the most useful in Tarchetypes. We use it constantly, and I'll describe how this works. So first, you want to define this data frame that has all those scenarios in your targets file, and I do that with this scenarios object here. Tar source, like usual, as well as loading the packages that you'll need to in order to define things. Then in the target list, that's where you would call tar map rep, which defines this very scaled, ambitious simulation over a bunch of different scenarios. And the basic structure is that you give it a base name, which I'm going to call simulations. You give it a command that's going to be repeated for each simulation, and that's just simulate trial. And I'm going to give it some parameters. I'm going to give it whatever symbols I need, just to say run the simulate trial function on whatever mean drug response we have, and on whatever sample size we have. And these parameters, and as well as any symbol in this command, are going to be substituted from values in this table right here. So we're going to run several simulations with mean response drug equal to 2, several with mean response drug equal to 1. And similarly for the sample size, each row of this scenario's data frame is going to correspond to a different kind of call to simulate trial for this target. There are only 4 rows to this scenarios table, but that doesn't mean we're only running 4 simulations. So each variant of call to simulate trial is actually going to run 1,000 times here, because we have this batch replication structure that's reflected in lines 26 and 27 here, where I have 25 batches. So that's 25 targets or dynamic targets for each simulation scenario. And each one of those batches is going to run 40 replications. And we do this batch replication because there's actually, if you have a large number of targets that are reasonably quick to run, then there's overhead generated for each individual target. And it's very often more efficient to batch a larger amount of work into a smaller number of targets. And that's why this batching happens. And it's done in a way that you can really hit a sweet spot in terms of how quickly your workflow runs, and which different pieces can be alternately rerun and skipped. And the Tarchetypes package does special things with random number generator seeds to make sure that each individual simulation has its own reproducible random seed. We have another target downstream of all that, which we declare in the normal way, to take all the simulations and just group them by scenario and summarize the proportion of simulated trials that declared efficacy. That's going to be our power and our type 1 error. This simulations target is implicitly all of the replications and all the targets defined up here aggregated. So whenever you mention a symbol like this downstream of, let's say, this tar map rep factory or dynamic branching, the aggregation is implicit, and this is quite nice. Other pipeline toolkits have a manual, have a requirement that the user explicitly do some kind of map step and then an explicit join step after that. In targets, the joins are implicit depending on how you write your code. And of course, we have the report at the very end, which just takes the results target we have here and visualizes it. So the branching structure that we end up with in our pipeline, each square here is a dynamic pattern target. And each of these actually stands for one or more dynamic branches, which are targets that are created while the pipeline, that are defined while the pipeline is running. And it's really useful to have this kind of pattern because you may not know just what kinds of targets you'll need until the pipeline is actually running. You may get some of that information from previous targets. That's the mechanism that's happening right here. And so each of these squares is actually 25 targets based on how we defined it. And each of those 25 dynamic branches is going to run 40 simulation replications. The simulations target down here is going to aggregate all of these 4 scenarios. Aggregation implicitly happens within each of these dynamic pattern targets, these squares here. There actually does need to be one more aggregation step after that. But all of these 6 targets upstream, those are implicitly created by the tar map rep function. And so even though you really only wrote 3 different target-like statements, you actually end up with 8 nodes in this graph. The tar make function runs the correct targets in the correct order like usual. The only difference now is that we have many more targets in our pipeline. But it still ran them all in the correct order, and it still stored the results. When we read the simulations, the aggregated simulations, we have a data frame like before with 1 row per simulation result and columns denoting not only the p-values, but the efficacy scenarios. Tar map rep function brings those pieces of metadata in nicely. And then you have information on the batching structure, the seed, and there's a nice grouping structure that'll help you do advanced things downstream if you want. You can read the results like usual, and this is what the report shows as well. So and we have operating characteristics in this nice 4x3 data frame here. So we have for each of the sample sizes and each of the efficacy scenarios, the proportion of studies that declared efficacy, and we see that the first 2 rows measure type 1 error because these are the null scenarios, and they're well below the 5% target that we required, and the power is a little less than 90% in the 700 patient scenario and a little more in the 800 patient scenario. And so we might recommend to bring these results to the team and say, we're making an initial recommendation that this trial have 800 patients under the current allocation scheme and other experimental conditions. And that might kick off more discussion, more planning, more simulation. But it's a good basis of quantitative evidence to go with, even if it isn't always the final result. And to answer one of the questions in the chat, what do the names and column arguments do for tar map rep? So the columns argument makes sure that the sample sizes, the sample size and efficacy fields, the various elements of the scenarios data frame that you may not be using in computations, but serve as metadata, that'll make sure that those are appended as columns in the simulation output here. I didn't write any code and as far as the simulation is concerned to append the efficacy and sample sizes, but that's from the columns argument of tar map rep. Likewise with the target names, that's where the names argument comes from. these names like simulation strong 700, the efficacy scenario strong, and the sample size 700 are automatically appended to these target names that denote the batches that are running. So that's why that's what the names does. The names controls the target names, whatever columns of the scenarios you want to append to target names to make them informative. And this columns field says what elements of the scenarios data frame here do you want to use? Do you want to append to the simulation results? And it's a choice here because in the scenarios data frame, you might have list columns that are a bit too complicated to append to simulation output, and maybe they're not amenable to converting to character strings, and so you might want to omit them as far as constructing informative target names. But you can select those with this nice tidy select syntax, like I've done here with the all of selectors. You can also choose to completely omit these, and that'll just create inscrutable hash suffixes for the target names here, and it'll omit these this metadata here. But it is quite useful to have. And in general, if I get a lot of questions asking, well, what if an individual simulation goes wrong and you have to troubleshoot it? How can you find out the conditions under which you ran that simulation and analysis? And my answer to that is that it's really good practice to append nice, neat metadata summaries next to your simulation output, next to your p-values for anything that you might want to track proactively, prospectively, when you're setting up a simulation. But there's also a lot of nice debugging functionality in the targets package. And the user manual in the debugging chapter has a lot of advice on how to debug simulations that sometimes fail with a cryptic error and that most of the time succeed. So those techniques of interactive debugging and saved workspaces are really going to help you. These pseudo-random number generator seeds are also going to help. It's a murky, tricky process sometimes, but there's a lot of techniques to troubleshoot, and the user manual is full of them in that chapter. All right, so we still have this problem of computation time, even after we've organized a nice pipeline with targets. We've scaled up to a large number of simulations. I would go more than just the 1,000 simulations per scenario that I showed here. But we did scale up to close to the level that we'll need. But there's something pretty big that's missing still, and that is parallel computing. For pipelines that take a long time to run, even if you're skipping results a lot of the time when they're up to date, you still have this problem of running those first pipeline runs to begin with, running all those targets for the first time. You're going to have to do it at least once, and it's really helpful to be able to automatically use something like distributed computing or local parallel computing to do that. And historically, targets and Drake before it have tried a bunch of different approaches to make this work. And targets is converging on something that's going to fit the use case, that does fit the use case extremely well. And it's going to scale to a lot of different backend technologies. And that's what Crew is all about. So before I'm going to go right into Crew for the last part of the presentation. But first I'll stop for a second and ask if there are any remaining questions before we go on. This is going to be a bit of a shift of gears here. And I just want to make sure that we're all on the same page before continuing. So if you want to type your questions into the chat, or if you want to go off mute and ask a question, that would feel free to do so. Question that I see in the chat right now is if I could clarify the difference between a batches versus reps. So when we go back to where we're defining this this batch replication structure in tar map rep, this is defining a given number of batches and a given number of reps. So a batch is, in this case, a single dynamic branch target. When you run tar make, when it says start branch simulations strong 700, etc., this is a single batch. And it's a batch because it contains multiple simulation reps. So this target is one step in the computation. It's a batch, but it's running several simulation replications behind the scenes. And if you were actually to do a tar read on this target, then it's actually going to have multiple rows where one row is a given simulation rep. So a batch doesn't always equal a scenario because we have 4 scenarios here, and the way we've defined it, we have 25 batches and batches per scenario. So what that means is this row here, the strong scenario with sample size 700, that's going to have 25 different targets. And each of those 25 different batch targets are going to have a bunch of reps. So this is a batch, and this is also a batch, but they're running the same under the same scenario. And in the way I've defined it, it's there are going to be 25 of those. So we can talk about that a bit more offline if you'd like. Other questions? Would I use targets during an exploration phase? That's an interesting problem because there are things that targets does to make the pipeline formal and declarative, to run it in automated fashion where the whole flow of the simulation project happens together. Everything happens in different processes. It may happen on different nodes on a cluster or on AWS batch. And those aren't really accessible by interactive means. And it also protects the computation from the user in a lot of ways that make the results reproducible. So the very mechanisms that make targets amenable to a trustworthy, scalable pipeline also are mechanisms that resist interactivity. And a lot of the debugging chapter describes ways to break out of to break into the pipeline and for the purposes of debugging and making things more interactive. So there's always going to be this set of trade-offs, but there are debugging techniques that make certain parts of the pipeline more interactive for debugging purposes. And I guess the other kind of interactivity is for exploration. For general exploration in the absence of bugs, I find it a really useful pattern to run just a few simulations to start out with, just a very small number. Read them with tar read, explore them, and use the nice data store to make sure that you don't have to worry about saving files that you want to explore yourself. That can be really convenient as far as that's concerned. And then when you're more confident in, let's say, one of the scenarios of the pipeline, you might want to run that pipeline. You might want to aggregate the results, and you might want to use these simulations for downstream analysis. And you can do a tar read on the simulations to get them into your R session, and then you can explore those interactively. And then for whatever analyses you want to run downstream that you've planned using this interactive exploration, you can then add more to the pipeline. So it can be very often a back and forth between running a pipeline up to where you've defined it, and then exploring those results, adding more targets to the pipeline, running it to get those results and exploring them. It's very iterative. I think I have time for one more question before I move on. And the chat, somebody in the chat says, could I speak to my experience with sharing targets code with other collaborators? So I think that it's targets pipelines because of the central data store, and because the current state of the results are so important for the sake of figuring out which results are up to date and which ones need to be rerun. It usually works best if there's one maintainer or a small number of maintainers for a given simulation project. If you're running the simulation code yourself, it's good to have a different data store. It usually works if you may have different branches of the code, but maybe separate data stores. Especially for cloud computing, if you have an S3 bucket, it'd be good to modify the prefix or the bucket to make sure that different collaborators aren't unintentionally overwriting each other's results. And it's a tricky process, but I think there's a lot of planning that's going to need to be involved as far as planning how you're going to do your workflow together. So we could talk about this a bit more later if we have more time. Have you found it's difficult to communicate your code, or do you have tips for doing so? I like to write documentation for functions. This is especially important for validating simulation projects. Internally in my company, we have a process for validating any and all code that we develop. It has to be trustworthy, and we have code reviews that are part of that. And I'll write documentation and examples for functions inline in the function source code that are reproducible, that are very much look and feel like package documentation. That really helps, as well as descriptions about what the function does and what it's all about. That really helps somebody who's validating your code and making sure it's correct. And in a regulated industry like ours, and for the high stakes projects that we work on, regardless of regulation, it's so critical that these simulation results be not only correct but transparently correct. And function documentation is really important. I've had colleagues who recommend writing a vignette that runs very simple cases of each of your simulation functions to demonstrate how they work, describe more about the trial, etc. Those can be in our Markdown vignette form outside the pipeline. Moving on, we have a half an hour left in the workshop. I'm going to get to Crew, which is a new package that it's one of my major preoccupations for this year. It's a really exciting set of developments that a bunch of targets users and I have been really looking forward to for a number of years, and it's finally here, and it connects with targets, and it really helps for simulation. So Crew is a bit of a lower level tool than targets. It is a utility package for asynchronous computing in R. And just to give you a flavor of the ecosystem that Crew sits into, probably the most famous package for this in R that does asynchronous distributed computing is Future, developed by Henrik Bengtsson. It's a really friendly framework that abstracts parallel computing. It's very easy to just set a backend technology and then have that code be portable from system to system. And I'd be remiss if I didn't mention the parallel package as well. It's in base R and it's been around for quite a long time. It's not on this list, but it is the most fundamental package in this ecosystem. ClusterMQ, which I and targets have been using for a long time. It's a fast functional programming framework for computing clusters, traditional clusters specifically. It's built on top of ZeroMQ. Mirai is my favorite package on this list because I develop on top of it for Crew, and it's done wonderful and amazing things. It's built on top of a messaging framework called a concurrency framework called NNG. It stands for a Nano Message Next Gen, and it's one of the successors to ZeroMQ. It's highly multi-threaded. It has asynchronous input and output, and makes this request reply network programming pattern incredibly reachable and useful. And Mirai is actually means future in Japanese. It's a next gen future, if you will. And so Crew is a package, like I said, that's developed on top of Mirai. Mirai is developed on top of NanoNext, and NanoNext is the NNG client for R. Mirai and NanoNext just do so many incredible things with network programming that are absolutely essential to what Crew does and to what Targets does. And Charlie, the maintainer of these packages, he's brilliant. He is an excellent C programmer, and he is we've worked closely over the last year to accomplish our shared goals for parallel computing in R, and the collaboration's been amazing. The packages are incredible, and one of the end products is Crew. Crew is actually it's a very simple interface compared to what Mirai and NanoNext do. So Crew is fast because Mirai is fast, and it's also frugal because it's one of the goals of Crew is to be able to auto scale semi-persistent workers as you run this kind of pipeline where initially the work surges, where you have a lot of simulations that you're running, and then the work subsides as those simulations complete and those results come in. And maybe the pipeline, the task load surges again if you have another phase of the pipeline to run a bunch of tasks, and together Crew will detect the demand for workers, the task load, and match that with the number of workers that are running or that are starting, and it makes an intelligent decision about how many new workers to launch. Each of those workers, those processes that could be running locally, could be running on AWS batch, could be running on Crew cluster on a cluster. That work, those processes that can run those workers are going to scale up, and each of those workers is going to do one or more tasks. And then, thanks to features in the Mirai package, when the workload diminishes as more tasks finish, you can configure Crew and by extension Mirai to relinquish those workers when the tasks are done. So, for example, it's really good practice to set a maximum idle time so that the workers will automatically shut themselves down if they've gone for enough time without doing any work. You can also set a maximum number of tasks. There's this really nice smooth continuum between the persistent workers of something like Cluster MQ and the transient workers of something like Future. And Targets has needed this balance for a long time, and it finally has it with Crew. Crew is also friendly because it has this nice central object-oriented interface to submit tasks and to collect the results. You have simple methods that'll describe to push tasks to the queue, pop them off the queue, and you don't need to keep track of individual future-like handler objects for individual tasks. You can forget about the task once it's pushed to the central controller, and all you need to do is go to the central controller to get the results back. And parallel programming in R has needed a task queue like this in package form for a long time. And that was a major motivator. It also supports different kinds of workers, different groups of workers with through this controller group mechanism. And it's seamless, and it's a way to define, for example, let's say you have a bunch of workers that run processes with 2 CPUs and 8 gigs of memory, and then you have a group of workers with a GPU and 4 CPUs and 16 gigs of memory, and you can submit different tasks to different controller groups, things that you've defined in advance. And you could have a controller group with local workers, and then a controller group with a controller with local workers, and a controller with AWS batch workers, and to have that work together in a single unified controller group. And best of all, there's this launcher plugin interface that allows users to define very easily define a crew extension for for your favorite distributed computing platform. And there's already launcher plugins for clusters. Somebody in the chat mentioned crew cluster, and also AWS batch, which is brand new. I just released it last week, and have just yesterday started running targets pipelines with it. And it's a really exciting development to get targets fully running on the cloud, not only for storage, but for compute as well. And Crew is designed in a way that fits really well, like I said, into targets, but also into Shiny apps. So the interface looks like this. So you load Crew to start. You create a controller object with a given maximum number of workers and some configuration settings. So this controller is going to use a maximum of 2 workers. It's not going to scale up beyond that amount, but it might scale up to that amount. The push method of the controller lets you submit a task asynchronously. So you'll give it a name and you'll give it some R code to run. And despite the fact that the command is to sleep for 10 seconds, you run this push method and control returns to you immediately, because the command is going to run asynchronously in some kind of background process, whether it's local here or it's on a distributed computing platform. And there are any number of ways to wait until a result is available. And when one is available, it returns this nice monad-like object that not only has the result in a list, but also metadata like optionally the name and the command if you allowed it to, if you configured the push call to keep track of it, as well as information about errors and warnings, and even a traceback if an error was thrown. So you can push and pop individual tasks through a loop like this. You might naturally find yourself in a loop like this when you're running a Shiny app. And you can use different plugins very easily. In the available plugins and in plugins that you create, a common pattern is to create a nice helper function to create a controller. We saw a crew controller local before. There's also from crew cluster, there are different controllers for different kinds of clusters. So this one is for a Sun and Grid Engine or SGE cluster. There's also plugins for Slurm, LSF, and PBS and Torque. You can give it all kinds of configuration settings for resources, for environment modules, etc. And then when you push a command like usual, it'll just submit the job to the cluster instead of running it locally. And here I've actually created a controller group with the local controller and the Sun Grid Engine controller, and selecting the right controller for the task just is a matter of specifying the controller name that you've given it in the name argument here. And like I said, plugins are one of the best things about it. Plugins they're actually, if you know even a little bit about R6 programming, plugins are actually really easy to write. All you need to know is the familiarity with the platform that you're programming on, and all you need to really do is to write a method to launch a worker and terminate a worker. So if I'm defining a local custom launcher class, for example, that runs workers with the process X package to run background processes, all I do is define a launch worker method and a terminate worker method. Launch worker is always going to have these arguments from call to instance, and terminate worker is always going to have this handle argument, which is just an object returned by launch worker. And all this is just starting a process that runs the R code in the call. That's just a string that runs a command in R. So all you need to do in launch worker is just, you already have code that runs a command in R to start the worker. All you need to do is create the computing environment that starts R and runs that task. It's very simple and very little left to do for the launcher plugin developer. Same thing with terminating worker. You have complete control of what this handle looks like because that's the return value of launch worker. And so as far as terminating the worker, that's very easy. And it's good practice to also write a helper to create that controller object. There's configuration details like everything from the worker behavior to TLS encryption, which I recommend that this be enabled for anything other than local process controllers. This mode equals automatic is TLS encryption with a one-time password. That certificate's automatically generated and it expires as soon as the controller exits. Charlie made this happen with an R consortium grant to develop NanoNext, and it's extremely helpful for just network security in our zero trust world today. But anyway, the controller creator here, it's not quite a constructor, but you can call it that if you want. It just involves creating what's called I actually call this client instead of router. I'll have to change that. And it'll create a client object, and it'll create a launcher object, and it'll create finally put those together for the controller. And the usage of plugins, it's exactly the same as you get everything that's in the controller: start, push, pop, terminate. All of that is the same from plugin to plugin. There's very little that needs to be done from a plugin developer standpoint. It took me as soon as I figured out just the features that I need to add to Crew to make cloud computing plugins for targets. It only took me about a day to write Crew AWS batch. And fortunately, I had the kind of security groups already that would make it run nicely in Lilly's up and coming cloud computing platforms. And that's really how easy it is to write plugins in Crew. And if you're interested, for anyone who's running Kubernetes for data analysis, if you're that far ahead in sort of in sophistication for cloud computing, let's talk about a Crew launcher plugin, and same goes for other cloud services too, beyond just AWS batch. Love to chat, and whether you write a plugin or I write a plugin, it'll be a really nice way to cash in on this functionality here. My colleague Daniel Woody wrote an early version of a Shiny app with Crew that uses Crew for asynchronous programming in the backend. And I've since adapted it into a vignette in the Crew package. And it really fits nicely into how people program Shiny apps. There's, of course, the future promises framework that's been around for a long time. That's for a bit of a different use case. That's for several instances of an app that are all sort of concurrently running on the same kind of platform. Crew is more meant for just apps with a large number of computationally intense tasks, and that everything that happens inside an app instance could be computationally expensive. And we see this in sort of enterprise apps that colleagues like Eric Nance have developed internally. He's very well known for enterprise Shiny apps internally and externally. And this is something that he's sort of trailblazed and explored, and this use case comes up quite a lot. I only demonstrate here with a very, very simple kind of app where we click a button to run a task, and that task runs in the background for a while. This timestamp increments every second and reports the number of tasks, and we get some nice computer-generated art at the end of a 5-second delay. So to start this app, I'm going to define, I'm going to write some packages in this app.r. I'm going to define a task that, again, runs for 5 seconds and uses the artsy package to randomly generate some art, a phylotaxis graph, a function to report the progress, and a really simple UI with a button and some outputs. After this, the server gets a little bit more involved, but it's still in the scheme of Shiny apps overall. It's really it's I guess intermediate level. It's not really that insurmountable. How I'm going to start this app is to create some reactive values, and these values are when they're called inside of a reactive expression, if the reactive value is changed somewhere else, the reactive expression that depends on it, every reactive expression that depends on it is going to automatically rerun. And this is sort of a backbone for this event-driven programming that you see a lot in Shiny. So I'm going to have the result, which is the artwork, and the status both be these render reactive expressions that depend on these reactive values I've defined previously. So if the reactive value in the plot automatically changes, then this result will automatically update and refresh on the screen. And likewise with the status. In the app, I'm going to define a controller, and the controller is going to start, and when the app stops, it's going to exit. And when the and now begins the interesting part. When you press the input button to submit a task, this run task function gets pushed as a job or as a task to the controller, and we can give it the data and packages it depends on, and then it'll set this reactive poll flag so that the Shiny app should keep checking for results while there are still results to collect. And then there's this observe statement here where we only run it if there are results that we're expecting. That's the purpose of this reactive poll reactive value. And invalidate later says that sort of after this, if we are polling, then we're going to poll every 10th of a second. We're going to check the result from the controller. If it's not null, which if there's no result to collect yet, if their result's still running, then this is going to return null. But if it's not null, then we update the reactive value with the plot, and we update the reactive status, and we revisit the decision to keep polling. That depends on whether the controller is empty. But because I've updated this reactive value, then that means that this render plot reactive expression is going to rerun. And so what that looks like is this app that you'll see at if you go to this shinyapps.io link, and there's also a vignette. In a targets pipeline, which is where all this comes together, crew and targets interact seamlessly. Targets depends on crew as an optional suggest package. When you use crew with targets, you have incredible power for distributed computing. And what you can do is you can have a complicated pipeline where targets automatically figures what to run and what tasks need to run right now, what tasks can be skipped, and what tasks need to depend on upstream things. So it'll automatically wait on upstream tasks that need to finish before continuing. And then once a task is ready, if all of its dependencies have been met, then targets will push a job to crew. Crew will do the auto scaling behind the scenes. And so what'll happen was you may start with a couple of initial targets, and then you'll run a bunch of simulations in parallel. Some might complete before others. The simulations that are ready to start, those will be submitted to crew. Crew will do the auto scaling on whatever backend you select, and will run things in parallel. Once those are done, in a pipeline like this, crew will automatically scale down if you supplied a sensible idle time to the controller. The simulations will aggregate, and the results will the operating characteristics will run after that because they depend on the simulations. They're not going to start before the simulations are ready. And then the report will run, and then the pipeline is all done. That's kind of what that is what that'll look like. And integration with crew and targets is as simple as setting a target option with tar option set. And this controller, you can supply any crew controller that you want to use, and use in terms of how you specify this controller. This is just ordinary crew at that point. So it's very seamless in respects the interfaces, the respective packages. And what this looks like in a pipeline, everything else in the pipeline is the same, except that we now have a local controller where we can scale to up to 4 workers at a time, not beyond, but we might scale up to 4 workers. And that if any one of those workers spends at least 30 seconds idling, then it'll shut itself down. And then if that worker is needed again, then crew will launch it from the beginning. And so this is a really nice continuum and really nice balance when it comes to scaling a computation up and down. And things like AWS batch, it takes a few minutes for a job to load. And so a perfectly persistent worker model isn't always the most convenient one. So you can make use of processes and jobs that are already running to run more tasks if more tasks need to be run, and then shut those workers down to save costs and resources. Whatever controller you want is a controller that you can supply. This is instead of a local controller, you could supply a Sun Grid Engine controller here. And you could also supply an AWS batch controller if the main process of the targets pipeline is in the same security group as the batch jobs themselves, and TCP connections are open within that security group, and we can talk about how to do that offline. Then you can use the AWS batch controller, under development and brand new, but still available, to run a targets pipeline on the cloud. And there's plenty of opportunity to write more plugins for crew. I don't think, as far as I know, it's few people have figured out how to best adapt Kubernetes to traditional cluster or Slurm-like workloads. But once that gets figured out, and somebody knows how to create a platform like that for statistical analysis, then it's going to be really exciting, and it'll take probably less than a day to write a Kubernetes plugin at that point. Once that knowledge is already and that infrastructure is there. So this is a really exciting time for cloud computing and targets and Shiny apps and in R in general, I think. And to recap what I've talked about, these slides are publicly available. There is the source of the slide deck is also available. There's example code to run this pipeline. Apologies that I don't have the link to it, but there is also an R and Pharma backed cloud workspace with that example code. If you'd like to run it, that project does exist. Thanks to Phil Bauscher and the other R Pharma organizers for having me and for making that infrastructure available. And the documentation for the specific tools is available and quite extensive. There's targets, there's documentation on targets with crew, on crew itself. Each of the packages with plugins like Crew AWS Batch and Crew Cluster also have their own sets of documentation. Lastly, I'd like to thank our OpenSci for not only adopting targets and target types and spreading the word, but also just helping me get my start as a developer of open source packages, and in general helping me start my career. such a technically astute and friendly group of people. They're an incredible community. Charlie Gao, who I've worked with and learned from extensively this past year, and without his this could have only happened with his support and his collaboration. There are features in Mirai NanoNext that he spent a lot of time on, that directly support Crew, and I'm extremely grateful for that. I had early contributions to Crew and Crew Cluster by a bunch of people as well. This plugin interface is really easy to get involved with. Also, thanks to our internal simulation team, we have a group of us working on simulation tools internally to design and simulate clinical trials. And also a lot more people who were major influences for this, and who I just generally look up to in the art community, and I'd like to thank them as well. And with that, I have time for more questions. Either in the chat or if you want to unmute yourself, that would either would be great. I see a question: is there any way of following messages, warnings, and progress from Crew workers when they're running a target? That's a bit of a tricky problem, and that's one that I've been thinking more about, especially in the cloud, because CloudWatch workers are a bit difficult to dig for those logs. I've so far I've had sort of a light touch approach where you don't really need to know much or do much with your specific computing backend in order to run or write a plugin. That is something I'm thinking about, though. And I think for now, a task could have its own logs in its return value, and the workers, to get those logs in either a platform independent way or just in general. It is something that I'd like to explore more. Are there any plans for making crash jobs inspectable by post-mortem debugging? I think that's gonna also be plugin dependent. I think that on clusters, I would consider this mostly a solved problem, because the Crew cluster package exposes arguments to specify where the log files go, and you can assign just a folder to that argument, and then each worker will write to its own log file. It's going to be a bit harder in the cloud, but I am mulling over sort of what the best approach to that would be. Tools like Nextflow have a way of automatically digging into error logs and such, but I don't know how much of that will be possible in something like Crew AWS Batch. But I think it is important to see what we can do. Let's see. As far as reloading the R environment from a crash, so targets can help with some of that with its debugging. So the way that targets keeps track of data and can write what are called workspace files to keep track of the everything that we need to recreate a target's runtime environment locally, so you can do troubleshooting, including dependencies and a random seed. I think that's going to be helpful. I see at least one person with their hand up. I think we have time for maybe one more quick question. Is there any way?
Oh, I was going to say, I think you got one in the chat box if you want to tackle that one.
Great. Is there any way of forcing just one branch to be obsolete without making the whole target obsolete? That's tricky. There are functions and targets that can invalidate individual steps of the pipeline that can either in the metadata or in the data store. Obviously, you can manually delete files from the data store. Individual branches are a bit tricky. because those are hard to expose at the interface level just because of how confusing that would be to users in the general case. Um, but yeah, tar delete should work for individual branches. I haven't tried in a while, but I have been developing the code as recently as today, and it does. I believe you can. Let me know if you can't do this, but I believe you can select an individual branch name for that too. I think we're coming up on time. Thank you so much to everyone who attended. Um, and thank you to our pharma for organizing this and for agreeing to post the recording afterwards. And I use these recordings for documentation for targets, and it's really exciting to have this one.
Will,
And
I believe all the thanks goes to you. Thank you so much for doing this and bringing such awesome information to the R Pharma community. It's always great to see all the awesome things that you're working on. And I know the community really appreciates the time and effort and how well organized you are with everything. And thank you so much. As Will said, we will be posting this on our RN Pharma YouTube channel, as well as Will probably be sharing it as well. Here is the link to that. One quick note is that we post these, unfortunately from us, in a couple months, just because there's a lot of effort and time that goes into editing them and getting the approvals. So just keep an eye out on our blog page, and we'll probably put an announcement out when the workshop videos go live. Also want to say that if you attended today and you'd like to get the credential for attending this workshop, please watch out for the form that will pop up in Zoom. It will ask you for your information so that we can send you the badge. If you don't see that, as always, just send an email to info@rnpharma.com or reach out via the contact page on our website, and we can add your information and get you sent the badge. So one last time, big round of applause for Will and all the amazing content here today around targets and crew and the rest of the packages that you support. Thank you to everybody that came on for the workshop today. And there is an hour left of the Quarto workshop that I'm going to jump over to. A few people have asked for the Zoom for that. If you stick around, I will give it to you here in the chat box. Otherwise, we've got workshops tomorrow that are kicking off. There's 3 tomorrow, and then the Pharmaverse Friday will be happening on Friday. Of course, the workshops and the conference then for next week as well. So with that, stay tuned, and we will see you hopefully tomorrow or next week. And I will go ahead and end the workshop for today. And thank you so much.